All right. Good afternoon, everyone. Can you guys hear me? Okay, good. Some of you have been here, so you have seen a big chunk of this talk, but there's going to be some new stuff compared to the talk that I gave yesterday, specifically for the American University in Cairo. But it's good to be back and to see so many familiar faces from ABIC. So the theme for this particular session is to talk about opportunities that are available to African scientists, programs from across the globe. And what I want to specifically talk about is the Advanced Imaging Center, or the AIC, at HHMI Janelia Research Campus, which is just outside of Washington, D.C. in the state of Virginia. And the reason why I'm telling you about this imaging center in the United States, when Janelia set this imaging center up in 2014, the mission is always to make Janelia develop pre-commercial instrument available to everybody in the world, which means also that this imaging center is not accessible to anyone in Janelia. So this is completely for the outside world. So nobody at Janelia is allowed to use this imaging center. So you have to think about this as your imaging center. So as I'm going through some of the technologies, I really invite you to think very, very actively whether or not some of the technologies that I'm talking about is applicable to your project, so that you can actually apply to come to the imaging center. All right, so this is basically the campus of Janelia. So let's start by something that basically affects everybody who is doing microscopy, which is resolution. So this is the molecule of green fluorescent protein, or GFP, and I'm sure everybody here has heard of GFP. And if you were to shrink that GFP molecule to as small as possible, if you were to take a picture of that molecule using even the highest conventional microscope, this is the image of that GFP molecule. So even if you were to tag this GFP protein to your protein of interest and take a picture, even though this is the size of your protein, your image is going to look like that. So this makes interpretation of biological data extremely complicated, right? Because in that radius of diffraction limit space, you can actually squeeze in thousands and thousands of molecules. So you have absolutely no idea where your molecule is located. So that is very problematic. And this problem was described more than 100 years ago by Ernst Abbe, and it was not pretty much overcome until the early 2000s. And in the 1990s, people started to think about how they can break this diffraction limit. And one of those guys is Eric Batzik. And his idea, he published this paper in 1994, basically describing the idea that if this is the image of a microtubule, you're going to look. And when you zoom into the image until you can see individual pixels, the microtubule actually does not look good, because you actually have no idea where the microtubule is located. But the idea is that if you can make those individual molecule blinks, right, then you localize it mathematically, and you can keep doing this, and all you have to do is to repeat that process 20, 30, 40,000 times and just let those molecules continue to blink. Then you will be able to make a very precise map of where those molecules are located. And that basically gives rise to one of the super resolution technique called single molecule localization, super resolution microscopy. And for that, he got a Nobel Prize in 2014. And there are several others, super resolution technique, where we're going to talk about. Now, as soon as Eric got the Nobel Prize, he kind of gave up on this idea. Eric, he decided to move on to something even more exciting. And then leaving the localization super resolution technique at this state. Even though we have achieved super resolution, this is super resolution in 2 dimensions. The problem is that obviously life is in 3D, so doing 2-dimensional imaging is not really satisfying, frankly misleading. And so we have to figure out a way to do super resolution in the 3rd dimension. The problem with doing super resolution in the 3rd dimension is adding super resolution to the 3rd dimension is actually technically significantly more challenging than the actual Nobel Prize work. And so his good friend, Harold Hess, continued to think about how to solve this problem. And As I mentioned yesterday, any good microscopist, when you run out of idea, you turn to astronomy because astronomers solve this problem way, way, way earlier than biologists or microscopists ever will. And so if you're an astronomer, you are very interested in calculating the distance between Earth and a celestial body such as a quasar. And what astronomers will do is they build radio telescope on each end of the planet. And then based on the difference in the time or the delta T that these two radio telescopes detect the same signal, they will be able to mathematically, through a process called interferometry, very accurately calculate where that the distance to that quasar. And Harold basically adopted this idea and created this very first instrument that I'm going to introduce to you called interferometry palm system, or IPALM. Obviously, this microscope looks extremely complicated, and it is. It actually has 37 on-off switches, which is insane. But as complicated as it looks, the underlying principle of how it works is actually quite elegantly simple. So your sample will be sandwiched in between the two cover slips, all right? And then when the molecule is excited, light from the molecule will be collected by both objective lenses. So this microscope has two objective lenses. And the light collected will be channeled into this diamond-shaped beam splitter. So this inverted diamond-shaped thing. And this is the enlarged image of that diamond-shaped beam splitter. So you see this three horizontal line. The first horizontal line is a 6633 beam splitter, which means that when light hit it, 1/3 of the light will go through, the other 2/3 of the light will be reflected, okay? And then the second horizontal line is a 50-50 beam splitter. Half of the light will be reflected, half of the light will be transmitted. And then the last horizontal line is a mirror, so all the light will be reflected. So far, so good? So if you do a simple calculation, you will come to the conclusion that when light emerges out of this beam splitter, exactly 1/3 of the light will hit camera 1, the other third will hit camera 2, the last third will hit camera 3. Everybody still with me? Now, if the molecule is dead center in between the two cover slips, the light will hit camera 1, 2, and 3 simultaneously. So there will be no phase shift. They arrive in phase. When you put those signals together, they will not interfere with one another because they're completely in phase. So far, so good? So, but as soon as that molecule is off-centered, even just by a little bit, you start to create interference of the signal when you try to put them together. The more off-centered that molecule is in the Z plane, the more interference you're going to create. And so from there, we can now calculate exactly where that molecule is located on the Z plane, okay? And this created a very interesting super resolution microscope where the Z resolution is actually better than the XY resolution. The XY resolution remains about 15 to 20 nanometer, but now the Z resolution is 10 to 12 nanometer. So it is an extremely accurate and extremely powerful super resolution microscope system. Now, many of you have learned about what a confocal microscope is. So you use a confocal microscope to do optical sectioning, where you can section your cell into multiple sections. This super resolution microscope can section protein complexes, single protein complexes, into multiple optical sections. So what you're looking, is it possible to lower light? Anyway, so what you're looking at here are 5 focal adhesion complexes. So this is basically where the cells use to attach the extracellular matrix. So you can imagine that the cell is about 4 or 5 times larger than this room right now. So as I play through the movie, hopefully you will actually see that we're, thank you, that's fine, that's fine. We give people the liberty to fall asleep. It is my challenge to keep them awake. Is everybody okay with this? Awesome, all right. So you can see these 5 focal adhesion complexes. Now, when I step through the movie, you will see that this microscope is now optically sectioned protein complexes into steps, all right? So, and then we're gonna tilt it over, and you can see that the red color, which is the paxillin, which is the protein that the cell used to interact with the extracellular matrix, is at the bottom, and then the KANK1, which is the signaling molecule, is further up into the cell. So from there, you can now really structurally, ultrastructurally dissect molecular complexes. Another example, this is a single bacterium. And when I turn it around, this is the first time ever somebody can actually differentiate between the inner membrane and the outer membrane of a single bacterium using not electron microscope, but light microscope. And that is the power of IPALM. And one of this example is this studies of piezo1, which is a sensor that actually sense tension in every single cell. The fact that if I pinch you, you feel it, other than the pain, your skin also feels it because every cell has this sensor. So this project was accepted by the AIC. And the funny story is that 2 weeks before this group arrived, the principal investigator got a Nobel Prize. So this is actually the protein that got him the Nobel Prize. So he obviously cannot show up. But his team showed up, and then we do super resolution microscope. And for the first time, we can actually see in response to tension, how this two protein basically is twisting and opening and closing. And then we now basically can calculate all these structures, the various conformational change in response to tension, purely using light microscopy, and no electron microscope is involved. Now, with this kind of super resolution technique, obviously it is awesome. You can see a lot of molecular detail, but there is no free lunch in microscopy. Every time you gain something, you pay a very, very huge price to gain that extra capability, right? Now, IPALM basically chase after this spatial resolution to the extreme. And this is what I call the golden tetrahedron of frustration. Every time you gain something, you immediately pull yourself from all the other important parameters for imaging. So IPALM, by achieving higher spatial resolution, doesn't have much of an imaging depth. You can see focal adhesions and things like this, but anything higher into the cell, you cannot see it. There is no temporal resolution to talk about. It takes about 1 hour to 2 hours to capture an image, if not longer. And photostability is also an issue pretty much in every super resolution microscope. And I want to sort of give you an idea of what is the cost of achieving super resolution when you are trying to get that very fine ultrastructure in your sample. So another Nobel Prize winning technique, both of them got the Nobel Prize at the same time, is the STAT microscope, which stands for stimulated emission depletion, that was developed by Stefan Hell in Germany. And this microscope achieves super resolution through a completely different process. It basically uses this donut-shaped depletion laser to basically suppress everything that is beyond the diffraction limit that you want to see. So basically, the resolution, as you can imagine, is achieved by the size of the donut hole, right? So the smaller the donut hole, the higher the resolution, right? Now, to make that donut hole as small as possible so that you achieve resolution, they have to pump in significant amount of laser power. How much laser power are we talking about? We're looking at about 10,000 to 10 million watt per centimeter square of power. And as I also mentioned yesterday, I don't want to let people just forget about 10 million watt per centimeter square of power. I actually want to give you an impression or an idea of what 10 million watt per centimeter square actually means. So let's say if you go to the Sahara Desert on the hottest day possible, it is so hot that you're about to melt, right? The sun is delivering to you at most 1 watt per centimeter square of power. By the time you get to 3 or 4 watt per centimeter square, you will be in emergency room. You are expecting a cell to take the power of 10 million watts, 10 million suns. This is absolutely insane way of doing imaging. So this is obviously not a healthy way. A lot of time, a lot of people have gotten a super resolution image of just spectacularly dying cells and nothing else. That's not how you want to do biology. And then the localization super resolution technique, the single point super resolution technique that I've mentioned earlier, does not fare any better. It is still extremely bright, extremely intense. In fact, confocal basically is also within the same range, still extremely high power laser. So a lot of time when you're imaging, you also see your cells die very, very quickly. And ironically, the only super resolution microscope that did not get a Nobel Prize because the guy died early. So if you want a Nobel Prize, don't die. And is the relatively the gentler of the three. By no means gentle. It's still, sometimes 10 to 100 power of the sun, but at least it is gentler. So that is the cost you pay. You will likely kill the cell simply because you want to chase after super resolution. So in the mad rush to gain higher and higher resolution, many people forget that just by adding contrast to your microscope, you can see a lot of biological details. Most biologists don't really care what is the down to what nanometer they can see. What all of us as biologists care is, can I see the biological event and the biological story that I'm trying to follow? And a lot of time, just by adding contrast to microscope, you can actually see all the biological details, and it's not always about super resolution. So one way to generate very good contrast is to make sure that you only excite the layer of the cell that you want to see, nothing above, nothing below, just that layer, right? So one way to do that is to shape your laser into the form of a sheet, and then you scan that sheet up and down. So that basically gives rise to what we will call the light sheet microscopy. I'm pretty sure all of you have heard of light sheet microscopy as well. Okay? All right, so this is basically what a light sheet microscope will do. Now, what you usually do on a light sheet microscope is that you always have more than one objective lenses. You gotta have at least two, preferably four. So in this case, this is a light sheet with 4 lenses. There are 2 excitation lenses where they bring in laser, and then there are 2 detection lenses where you can actually see the image, right? So this movie is actually quite old by microscopy standard. This is generated in 2013, also at Janelia. So this is 12 years ago. So this is the first time anybody has ever seen neuronal activity in a live animal. So you are literally looking at neuron firing inside the zebrafish embryo. Some of you may have seen this movie. Quite a lot of people stole this movie in their seminars. So at one point, you will also see a burst of activity. We have no idea, see that? And we have no idea what that zebrafish is thinking, probably food, but that's okay. So we're still trying to decode what that neuronal activities are doing. But this is a very important leap forward because you can now see neuronal activity in a live animal. And at the same time, you can now also track the movement of every single cell, for example, in a developing Drosophila embryo, right? Again, this is the power of light sheet microscope. It is so gentle that the cells are totally fine. The animal is fine without a problem. They can undergo development sometimes to full organism. Now, the problem is if you're using zebrafish, if you're using Drosophila, you're fine. But if you're studying larger animal like mice, then you have a different kind of problem. When mammalian system develop, skin start to develop, blood vessels start to develop, body cavity, hair, all these things will start to develop, and every single one of these biological features will absolutely mess up your imaging because they create heterogeneity in the refractive index, right? So the photon got bounced all over before it even come out of the biological system. So this is a very big problem when you're trying to study larger organism. So we have to create a microscope where it can automatically assess the quality of the image without human intervention, because if you're trying to follow a mouse develop, it could be days. You're not going to sit in front of a microscope for days, right? So all this microscope can automatically detect if there is some changes that needs to be made, and it will make those changes. The light sheet actually have 12 degrees of freedom. It can pitch, it can yaw, it can tilt, it can automatically adjust its own intensity so that it guarantee the quality of the image. And also, as the mouse embryo is developing, at some point muscle also develops. And when muscle develop, the embryo is going to roll away. So the microscope has to have the ability to chase after that developing embryo, even in the middle of the night. And with this kind of microscope, the very first movie ever that basically captured a mouse embryo at the cellular level was generated. And so this gives rise to this simultaneous multi-view or sim-view light sheet system. And this is the mouse developing movie. So now you can actually see that embryo basically track the movement of every single cell, and you can actually start to see the notochord developing. On this side, you also start to see the correction of that microscope kicking in. As that embryo gets bigger and bigger, all the correction starts to kick in. The microscope gets busier and busier. In fact, if we turn off this software, which we like to call autopilot, this mouse embryo would have rolled out of this room, probably rolled out of this building. So actually, this microscope is chasing after it on the stage so that it keeps that entire embryo completely centered in the field of view. And this is another example. This is the endothelial cells, all in pink. At some point, endothelial cells will differentiate and then become cardiomyocyte. And when that happens, what we do is we flux out a gene, and then the endothelial cells that have developed into cardiomyocyte will turn green. So as I play the movie, you're going to start to see the emergence of more and more and more green cells, and that basically shows you that the cardiomyocyte they have differentiated into cardiomyocyte. So you're looking at differentiation happening, and then this is the cardiomyocyte channel. Actually, this is more interesting, more informative. So now you can actually see the first pair of the heart chamber starts to show up, the second pair follows, and then the heart starts to beat. All basically you are able now to follow from differentiation all the way to organ development. Now, we didn't stop there. So you guys are the second audience in the world to ever hear this. The first audience was yesterday. We are in the process of developing another instrument, another capability to add to the SimView light sheet system. I'm going to give you guys a little bit of a background of what this new capability is doing. So when you're using fluorescent microscope, you're excited with a shorter light, and then, for example, you excite the light with 488, and then the green light will emit, right? If you turn off the laser, fluorescent emission will drop. Now, when you turn off the laser, the emission will drop, but it always follows a certain decay curve. And that decay curve is very, very sensitive to the molecular environment of that molecule. So every molecule can always have a decay curve, usually in a nanosecond. Some of them are even in the picosecond range. Even autofluorescence molecule like hemoglobin and everything will always have a decay curve. So if we can actually follow the decay curve and not the actual emission, we can actually tease out a lot of information. We can even do a lot of fluorescent imaging without your sample being labeled with any fluorescent molecule, and that is called fluorescent lifetime imaging. And so this is the original SimView system. This picture does not have all the unnecessary component. It is actually more complicated than this, but this is the essential component of the SimView light sheet system. And then so what we're gonna do now is we basically add a pulse laser on the side, and that is brought in, and through a power lens, we now spread it into a light sheet, and then it is now combined into the original light sheet light path. And then this is the camera at the bottom. So instead of going to the camera, we slide this periscope in. Now the light got reflected to this side, and where we can actually put in a spat array. This is the array that will allow us to detect lifetime imaging rather than emission, just pure fluorescence emission. So this particular capability will be announced in the next round of proposal call that will come out in February. So I'm going to show you, this is fresh out of the oven. So this is a zebrafish, a very small corner of a zebrafish. This entire zebrafish has no fluorescent label. So this zebrafish is just a pure wild-type zebrafish. And what I can show you based on just the autofluorescence and the lifetime from 1 nanosecond to 4 nanosecond, we can now actually look at the endothelial cells in blue, and then also the neutrophils in pink, just purely from the lifetime autofluorescence signal in this completely unlabeled zebrafish. And so you can imagine that now you can either do a lot of multiplexing, basically a lot of fluorescent color in a single sample, or you can also ask the questions, does my molecule has now moved into a different environment where it may have changed its function? So this is something to think about for everyone in the audience. Now, all the light sheet system that I've described up to this point uses what we will call a Gaussian light sheet system. So the light sheet is very, very thick. Sometimes it's almost as thick as 1 micron. Almost as thick as 1 micron. And so, sometimes 2 or 3 microns. So those light sheet is good for large sample like a zebrafish or Drosophila or even mice. But what is really bad is the cell biology. The light sheet is almost as thick as the cell, if not thicker. So if your light sheet is thicker than the cell, then you cannot do optical sectioning, right? So you cannot really you need that light sheet to be as thin as possible. And so we have developed also another such instrument. This was developed by Eric Betzig again. This is after he has won the Nobel Prize. And so this is the lattice light sheet microscope. This system is now actually commercialized. So this system will soon be decommissioned from the Advanced Imaging Center. So this is now a very, very thin light sheet specifically designed for cell biologists. And I'll show you one of the examples. So this is basically an interaction between a T cell and then an antigen presenting cell in blue. So this is the first time anybody has ever seen how these two cells interact and how immunological synapse, you can see a lot of finger-like protrusions from the T cells that is probing the surface of the antigen presenting cells, literally trying to pull those antigenic material off the surface of the antigen presenting cells. And so this is a very important leap also for anybody, especially immunologists, to be able to see this for the first time. And then this is a similar event. This is from Lakshmi at the NIH. I come to Janelia to use the lattice light sheet in the early days. When I play the movie, you're going to see these cells climb from here to there and touch the antigen presenting cell. This is the antigen presenting cells. This is the T cells. Before they touch, you do not see any yellow. Yellow basically shows the colocalization between one of the T cell receptor complex component and the very, very downstream signaling kinase that basically tell you that the T cell receptor complex activation has completed. So when you actually see yellow, you're actually looking at T cell activation, T cell receptor activation as they interact with the antigen presenting cells. So come up, there is no yellow, touches the antigen presenting cells. Now the T cell receptor activation is completed. And then this is the exact same event. I would like to invite you to imagine that you are the antigen presenting cells, and now the T cells come and form T cell receptor complex in your face. Okay, here it is. Now you see the formation of T cell receptor complex as it is probing your face. All right, here is another example. So this is the back of a Drosophila embryo. At some point during the Drosophila development, there will be a dorsal closure where the abdomen starts to fold. And so I want to show you the capability of lattice light sheet. So you can clearly see a lot of cells are jiggling around. We're going to pause that movie just to show you how lattice light sheets scan through your sample. This is slowed down by almost 1,000-fold. And then it continues. Now, for those of you who have used confocal microscope, you will be very happy if you can generate 30 stacks, 40 stacks of images, right? And I'll let you count, and I'll let you see what is the end results of how many Z stack or Z stack we have captured. It is close to 800 something. So this organism does not show any sign of stress simply because the light sheet is extremely gentle. The record at the Advanced Imaging Center is 10,000 stacks from one single specimen, and that's how long and how many events we can potentially follow. And I want to specifically talk about why this particular microscope is so gentle. The reason why it is so gentle is because the light sheet is extremely thin, right? We have talked about that. And when you excite only a very, very thin layer of specimen, you have very, very good contrast because nothing above and below it are fluorescing. So you see only that very, very thin layer. And you see a lot of biological details. Guess what? The moment you see biological detail, you automatically tune down the laser power because you don't need that much power to generate the detail. And that is give rise to probably until today, the gentlest live cell imaging system on Earth. Now, just to prove my point, for those of you who also study cytokinesis or cell division, you will also know that this cell division is probably the most light-sensitive biological event. If you shine too much light onto the dividing cells, they will not divide. In fact, they will get arrested in metaphase and they will never come out of it. And I'm going to show you how many rounds of cytokinesis or cell division this Drosophila embryo is going through. And this is a synchronized cell division during the syncytial stage of Drosophila development. And it will just keep going on and on and on. And I never thought I would say that, but after a while we say, wow, this is getting boring. Let's stop it. But actually, this is a pilot study. This is not what this group from Montpellier comes to AIC to do. What they want to do is something even more challenging, is to see whether or not they can follow gene expression, basically at the molecular biology level, from gene expression all the way to the cellular behavior inside a living organism. So when I play this movie, you're going to see one round of synchronized division. This is basically the last round of synchronized cell division. And then what happened next is that the appearance of pink dots, right? The pink dots basically mark the site where gene expression, specific gene expression is occurring that translate into a protein that will affect the cellular behavior that force the cells to undergo dorsal closure in the developing embryo. So you are seeing gene expression. Now you see that the cellular behavior has changed, especially for those that express. And those that do not express do not fold. So now, with this kind of microscope, you can now follow everything from molecular biology all the way to cell biology, all the way to developmental biology in one single experiment. All right, so this is all nice and good, but there's always problem. That's why we have job security. So if you want to go deeper and deeper and deeper into the specimen, you run into a different kind of problem, which is light aberration and light scattering. I'm not sure if you ever see this event in Egypt, but that's okay. So if you're in, like Seattle, where it rains every day, you will see that when you're driving, you're going to see light aberration, you're going to see light scattering. This happens on a daily basis. We see it all the time. And when you go deeper and deeper into the tissue, even though you can excite that molecule, the photon is having a problem coming back out of that specimen. And even when it comes back out, you have no idea whether it is generated in the first place, because it has been bounced around into the organism for so much. And so we have to solve this problem, and very, very difficult problem to solve. And in order to solve that problem, again, we have to turn to astronomy because they actually have solved it. So basically, if you have an earthbound telescope and you want to look at the stars in the distance, and the light coming from the stars has to fight through the atmosphere to get to the telescope, right? Or your eye. And that's why a star twinkles. And it goes through all the atmosphere, the pollution and everything in the atmosphere that messed up the star's light. And so that is not good for astronomers. And so what they will have to do is to solve that problem. And what they do is they fire a very powerful laser about 90 kilometers up into the sky. And where the laser encounter a sodium layer that that naturally occurring in the sky. And when sodium see the laser, it fluoresces. So what you're doing there is that you create a guide star in the sky that is actually not there, right? That is just artificial star. But the light coming from the star has to fight through the same problem to get to the telescope. So the wavefront is also distorted. So to correct that, you create a counter wave, equal amplitude but opposite direction. And you can do that with a deformable mirror. So basically, you just create a counter wave. And then when the wavefront, distorted wavefront encounter the counter wave, everything is straightened out. So now you can very quickly and very easily, frankly, correct for the distortion of the light. And that basically is the underlying principle of adaptive optics. Now, you can put adaptive optics into a microscope, And this is an example. So this is a zebrafish embryo. What we're going to do now is to build up the entire brain of every single neuron in the brain of a Drosophila embryo. Here we go. Now we're going in. You may get motion sickness, but that's okay. So now we're inside the brain. And what I'm going to do now is to turn off the adaptive optics to show you what the reality actually is. It's bad. It's really, really bad. So you have to correct for the distortion. Otherwise, you cannot even do neuron tracing, right? Simple neuron tracing you can't do. So adaptive optics is extremely important. And I want to show you another very important message, I guess. This is a notochord of a zebrafish, basically in the vertebrate. And as we peel back of the excitation wavefront, you see all this potato chips looking thing. This potato chips looking thing is the distorted wavefront as the laser is going into the sample. So even the laser itself is distorted by the very biological specimen that you are trying to image. And then again, if the detection wavefront is just equally as bad. So next time when you hear, well, you know, seeing is believing, you absolutely have to question, can you believe anything you see? It is very important, and there is a joke here that Eric likes to make, is every microscope is like a sausage factory. Everybody loves what comes out of it, and nobody wants to know what is going on inside it. It's a lot of unsavory thing going on in the sausage factory or a microscope. But since we have all this information, we can now absolutely correct for it, right? So this is the microscope that we have developed. It has a lot of modalities. I'm not going to go through it. And all of them has adaptive optics. So this is the blueprint of the microscope. And the very first thing is the configuration of the objective lens, which you're going to see the very conventional classic lattice light sheet configuration system. And then at the bottom of the sample, you also have another objective lens. And then on the other side, you can even do 2-photon microscope. You can actually put a rabbit down there. You can do 2-photon microscope with adaptive optics. Top floor is the detection plane. The floor below is the excitation path. And then the ground floor is the housing for all the laser. You can see the 2-photon laser and then all the visible laser all in one. So basically, this microscope has 3 different stories. So this is a system called which we call MOSAIC, which stands for Multimodal Optical System with adaptive imaging correction. So what kind of movie can you generate out of this? So this is a movie that actually was selected by National Geographic's magazine. It was put on the magazine and website for quite a while. This space here is basically the lymphatic blood vessel between the ear and the brain of a zebrafish. And what you're seeing here is the endothelial wall. And this is before the adaptive optics correction. This is after the adaptive optics correction. Now you see the entire wall of the endothelial cells and some junks that macrophages like to eat. And here they are coming into play, or actually immunosurveillance live. Now, another very interesting thing is this little spot in the endothelial wall is that every single transendothelial migration go through this particular spot. Something that we actually all missed until we showed this movie to a leukocyte biologist, and he almost passed out because there was this lifelong hypothesis that he has never been able to prove. So the endothelial wall is not created equal. They are very specific magical spot where endothelial cells actually allow transendothelial migration to happen. If you don't image something like this in a living organism, you will never be able to even capture this event. So this is, I think, a very important lesson for all the instrument developers out there, is when you finally develop a microscope where the excitation wavelength is so gentle that it basically matches the condition under which life evolves. Now you see the magic of life happen in front of you. And we have to go gentle. We cannot just go, you know, powerful all the time. The gentler, the better it is for biology. And I also think that with this kind of microscope, We have now ushered in an era where we have now realized the century-old dreams of cell biologists to be able to do cell biology in the context of a whole organism. Just to give you an example, this is a very complicated chunk of tissue in the forehead of the zebrafish. We pick it simply because it is complicated. And so we're going to pull all the cells apart. And you can clearly see that some of the cells are actually undergoing cell division in an animal, which is pretty cool. And what we're going to do is to focus on this cell. And hopefully I can convince you that we now have the tool to be able to track Golgi, ER, and mitochondria simultaneously in a living cell, in a living organism. So in fact, this is the very first attempt. We are now doing better and better at it. So as we are following this cell, again, in the living organism, this cell actually divides as we are following it. Here it is, it's about to divide. Boom, it has just divided. And so now we can do this kind of cell biology in the context of a whole organism, which is a very giant leap forward. Now, we're not done. So I'm now going to take you to the dark side of imaging where no photon is involved. We're now going into electron microscopy. Now, many of you know what electron microscope is, so I'm not going to go into details. So there are two kinds of electron microscope in general. There's transmission electron microscope, and then there is scanning electron microscope, right, or SEM. If in the good old days, if you are going to do volumetric EM, you basically have to rely on a microtome that very skillfully slice it very, very thin so that you create a ribbon of all interconnected tissue so that you can scan each one of them and then you put it together. And you always hope that while you're slicing this that you don't sneeze, because the moment you sneeze, you snot all over. So that is a horrible way of doing science. And it is very, very slow, and image registration is very difficult. So there are now two ways, very efficient way to do volumetric EM. The first one is called serial block phase microscopy, and it's basically a knife. It comes in like a salami slicer. It slices a very thin layer away, and then the scanning electron microscope at the top scan the image, and then the knife come in and slice it again, take an image, slice it again, take an image, right? So this can go very, very fast. But the Z resolution is not good because you are relying on an actual knife to slice it. And so when you put it together, the Z resolution always looks pretty terrible. A better way to do volumetric imaging with very, very high and near isotropic resolution is what we'll call using a focused ion beam SEM. So instead of a knife, a focused ion beam, basically a gallium beam, comes in and very accurately and uniformly mill away just a very thin layer of the sample. Now, how thin are we talking about? We can routinely mill down to 4 nanometer resolution. So the milling accuracy is 4 nanometer. And for those still thinking about what 4 nanometer feels like, that is the size of half of a large molecule. So we can cut a molecule in half if we want to. And that is the resolution, the cutting resolution of focused ion beam SEM. Now, there are many, many commercially available focused ion beam SEM available in the market. Now, I also mentioned to you that AIC does not provide commercial system. It is only pre-commercial system. And the reason why we have this is because most of the commercial system are only stable for about 3 or 4 days as it is imaging. So if you have a very large sample, sometimes it takes weeks, if not months, to mill through those samples. And commercial systems simply cannot do that because after 2 or 3 or 4 days, it becomes unstable. So it has always been Janelia's goal to be able to mill a very large sample continuously for a long time. So we have to create a very stable system. And this microscope that I'm going to talk about can continuously mill for a very long time. And the record at Janelia is 16 months of nonstop imaging. It's just mill and image for 16 months. And I'll show you some of the data that comes out of it. And so this system, again, through AIC is available to you. And I want to specifically show you things that are actually quite relevant to the scientific community here. This is a group of trypanosomes, Brucei, that we are going to mill through. And one of the current model is the trypanosome surface is basically caged by parallel microtubule cable, and nobody knows whether or not the microtubule actually start from the mouth, go all the way to the tail, or there is microtubules that are not connected to either end. So what I'm going to show you is this is the milling through the trypanosomes, and this is the trypanosome. And now you see the microtubule cage from head to tail. And then I want to show you a few. This is one of them where it actually does not touch either the head or the tail. So in single experiment, we solved the problem. You see these 9 structures of the cilia. Now it turned into 9 plus 2. At some point from 9, it turned into 9 plus 2, and the 9 plus 2 continue. So this is, we can image something like this pretty routinely, so that is important. And here's another very interesting example. This is basically an immune cells attacking a cancer cell. So obviously, this guy is picking a fight with a guy which is much bigger than he is. So now you can also see the exquisite detail of all the intracellular features as we mill through the sample. And I want to show you the interface where the 2 cells actually interact. So here's the T cells, here is the cancer cells, and we're going to make the T cell disappear. Look at the damage that it is doing to the cancer cells, right? So there's a lot of just massive damage onto the plasma membrane and the cytoplasm. And when we look at this image, originally we thought that this is just a physical attack where the T cells is punching the hell out of the cancer cells. But when we look at the T cell surface, it is completely smooth. So all the damage that you see is basically the results of chemical warfare. That is basically the lytic granules that is released by the T cells that created all this damage onto the cancer cells. And even that, as in this very, very lethal event, you can still see the exquisite, beautiful details of all the organelles inside the T cells, and you can absolutely also see the beautiful internal structures of the cancer cell. It's kind of a philosophical moment because you can still see all these beautiful structures of mitochondria inside the cancer cells. The cancer cells does not even know that it is dying, but the dying process has begun, right? And we're quite lucky to actually capture this particular interaction at this moment. And here is something that is also, again, relevant to the scientific community here, basically malaria. Many people have imaged malaria. You can see images and videos of malaria and the plasmodium in the literature all the time, but nobody has ever imaged malaria in the mosquito gut before. So we actually mail through the mosquito until we reach the oocyst of the plasmodium. And here it is, the creepiest image we have ever generated. And it actually has a surrounding cyst, and inside are all the plasmodium packed together like sardines, right? And every single one of them will make you sick, and that is basically how the plasmodium are packed together inside the oocyst. But this data is so information rich that I actually cannot show you all the information in one single movie because everyone will be overwhelmed. And so I'm going to show you, again, the other things that we can fish out of this 3D volumetric images. And this is basically the inner membrane complex of every single oocyst or the plasmodium inside the oocyst. Basically, every single one of them has a home inside that community. So all these are basically is in membrane. They're wrapped around every single plasmodium. And then what we can also show you as we zoom in is the apicoplast as well as the mitochondria that supplies energy and everything to this colony of plasmodium. So basically the mitochondria is in orange, and then the apicoplast is in green. All right. Now, in addition to doing EM, we can also do correlative light EM microscopy. And what makes our what makes our light microscopy part unique in this particular case is that the super resolution microscope is actually performed at cryo temperature. So we can actually very quickly high pressure freeze the sample so that it stays as biologically relevant as possible, and then do a super resolution image, and then go through free substitution and then embed and then we start the mill at the EM level. So this group from Pennsylvania State University came, and they are very interested in looking at cell-cell junction. So this is a plate, a plastic plate with a lot of cells sitting on it. And what they're interested in is, for example, they pick an area where these two cells interact. So we zoom in. This is regular conventional microscope. And then this is the super resolution image, again, at cryo temperature. And after we have captured that, we embed it, and then we start to mill. And as soon as we start milling, we start to make discovery. You can actually see this cell-cell junction there. And right away, we notice that ER is interacting with the keratin intermediate filament inside the cell. And as we mill in further, we also see ER making contact with the desmosome, which is the adhesion molecule. And we see all sorts of connection between this organelle keratin cytoskeletal network as well as the desmosomes cell-cell interaction, cell-cell adhesion site. And that basically, after we have traced everything, we realize that we have just discovered a tripartite regulatory structures that involve this three component of the cells: cytoskeleton, organelle, as well as adhesion complexes. So that was a pretty interesting discovery that has led to another publication. So now that ends the science part of the talk. Now I'm going to start to talk about the programmatic part, just so you know how you can get yourself to Janelia. We are not far from Washington, D.C. Ignore that. I know this is a mess right now. This is the Dallas International Airport, which is the Washington, the international airport for Washington. There is actually a direct flight from many African city, including Cape Town, Johannesburg, Lagos, and even Cairo that has direct flight to this airport. And Janelia is only about 10, 15 minutes away from the airport. And that is Janelia campus. Most of the research is happening in this landscape building. There is a Janelia Hotel, and there's also Janelia townhouses, and then this is the housing for employees. So when you are there, you will either stay at a hotel or stay at a studio apartment, and you are just steps away from the lab. Now, what makes the AIC program unique is that you actually don't pay for anything. If your proposal is accepted, we will cover the cost of your experiment within reason. So basically, you don't have to worry about, and you also don't have to worry about lodging, because lodging is completely covered. What you will have to do is to think about travel costs. There are a lot of travel grants out there. We also, in some limited cases, we also provide travel assistance if your proposal is accepted. So that is determined on a case-by-case basis. All right, so that is important to know because I think that is a pretty generous program. And this is basically the origin of all the proposal that has come to the AIC. We can certainly use more proposal coming from Africa. All right, so AIC is significantly more than just an imaging center, because this is a conduit through which Janelia, or the Howard Hughes Medical Institute, basically throw its entire institutional support towards your project. Before you even write a proposal, you should absolutely talk to us. The acceptance rate for the AIC is very high. It's about 45, 50%. So that is very, very high by any standard. But if you don't talk to us and you just write a proposal and submit it, your failure rate is about 95%. So talk to us. And talk to us early. Don't talk to us like the day before the deadline. Talk to us months ahead of time, and we can actually guide you through the process. We actually listen to your central questions. We help you with the experimental design. We even help you formulate your specific aim so that it matches with the capability of the instrument. And if you do it all in advance, we will even read your draft, give you correction before you submit. How about that? And if your animal require animal protocol, we will deal with it. We will help you with travel arrangement. For those who qualify for the visa waiver, we will help you with the visa waiver or help you with visa application. Before you arrive, your sample will be shipped to Janelia. It will be received. It will be well taken care of. Lodging is arranged for you. All the model organisms and in the vivarium, in the Drosophila shared resources are all available to our visitors. You can certainly ship yours. However, let's say if your mouse model is available commercially, all you have to do is to tell us the catalog number, for example, from the Jackson lab. We will be able to order the mouse and have it directly shipped to Janelia. You don't have to worry about shipping animal. And if you work with Drosophila, Janelia has the Bloomington collection, which is basically about 54,000 Drosophila lines available at Janelia. Also, thousands and thousands of zebrafish models. They're all there. And so all those are available to you for free if it matches your experiment. But a lot of time, most people actually ship their sample because they have make specific lines, which is actually quite important. And you should absolutely have used those organisms before you propose an experiment. Before you arrive, we will thaw out the cell for you, get it going so that you don't have to sit there and wait for the cells to thaw and recover. And if your project, for example, require primary neuron or any primary cells, the histology facility will isolate the cells for you from mouse or whatever, and get it going before you arrive. We also have about 5,000 cDNA that are fluorescently labeled, so we can pretty quickly rescue your sample should your construct don't work. And then there are also a lot of imaging probes. There are halo-tag probes. There are also a lot of calcium sensors. For those of you who are neuroscientists in the audience, you probably have heard of GCAM6 and all those. They are all developed at Janelia. So all those probes are available to you. Microscope time is completely free and is available to you 24/7. We don't schedule two competing groups on the same instrument. So if you are there, the microscope is yours. Some people who are from the United States actually bring in two shifts of people. So one shift image it during the day, the other shift image it during the night. We actually don't like to see microscope go dark. So please take a full advantage for it. And like I said yesterday, since you guys are jet lagged and sleep is overrated, you're fine, just keep imaging. And then during office hour, the application scientist time are also dedicated to you. Our application scientists don't entertain two groups simultaneously. So their time is completely yours. So they pay full attention to your project. And even after you leave, image processing and analysis support continues until hopefully you get that thing published. So I'm very proud to say that in about close to now 12 years of operation, the AIC has been able to maintain nearly 85% success rate. All right, call for proposal. We announce call for proposal about twice a year, probably about every 7 months or so. The next one will be announced in February with a deadline of April. So believe it or not, now it's actually a very, very good time to talk to us, so that if we give you recommendation, if we tell you, oh, you need to swap out this GFP molecule and change to this one, or if you make a new line, this is a good time to talk to us. The proposal is pretty easy to write. It is only about 1,000 words. And then it will go through 2 tiers of review. The first tier is done by the AIC team. We ask whether or not there are central questions in your proposal can be addressed by commercial instrument. If it can, then you kind of weaken the justification to come to the AIC. And then can the AIC instrument actually delivers the expected results? Sometimes people propose something that is so blatantly crazy that our instrument actually cannot deliver the results. We can handle up to biosafety level 2. Anything more we cannot handle, and you probably cannot ship it easily anyway. If your proposal passed the first tier, then it will go to the second tier, then it will be reviewed more like a grant. Your scientific rationale, does your experimental questions justifies the AIC instrument? Did you discuss any potential problems and alternative approaches? Bear in mind that you are going to be there for only 2 to 3 weeks. So everything has to be planned down to the last detail so that you maximize the use of the microscope rather than sitting there troubleshooting for days at a wet bench and never really touch the microscope. And quantitative approach is important. This is where a lot of proposals fail. We really want to know, what are you going to do with the data? Imagine that we give you a folder full of your dream data. What are you going to do about it? Do you know what to quantify? That is very important. You don't need to know how to quantify. you need to know what to quantify. And the amount of time we don't cap, but usually 2 to 3, sometimes 4 weeks. We obviously prefer people with a little bit of microscopy experience. So definitely don't send undergraduate who has never touched a microscope before. So I will leave my talk here. You can take a picture of the slide with a website as well as the email. If you send us an email, somebody will respond within 24 hours, and then we'll set up a time to chat with you and discuss your experiment. Thank you. Everybody can wake up now.